My name is Amy Rosenzweig, and I'm a professor in the departments of molecular biosciences and chemistry. I am thrilled to be presenting Joanne Stubbe to receive an honorary doctorate of science from Northwestern University. Joanne is a chemist and biologist whose research has focused on elucidating the molecular mechanisms of complex and important enzymes. She is best known for her work on ribonucleotide reductases, enzymes that provide the building blocks for DNA replication in all living cells. Joanne not only discovered how these enzymes use reactive free radicals to achieve difficult chemical transformations, but also contributed to the development of cancer therapeutics targeting these enzymes. Her contributions to science have been recognized at the highest levels, including the National Medal of Science and the American Chemical Society Priestley Medal, the highest honor bestowed by this society. Joanne's greatest contribution, however, may be the generations of scientists she has taught in the classroom, trained in the laboratory, and mentored over the years. Joanne became the first tenured female professor of chemistry at MIT in 1987, just before I started graduate school in that department. I worked in a different group, but also on enzyme chemistry. Joanne was a member of my thesis committee, and I, like many of her trainees, was initially terrified by her intensity and passion for science. I managed to graduate and have been lucky enough to collaborate with Joanne on several projects as an independent investigator here at Northwestern. I am just one of many, many female scientists inspired and uplifted by Joanne's leadership and advocacy. Her incredibly high standards and tenacious pursuit of knowledge have rubbed off on numerous students, postdoctoral fellows, and younger faculty of diverse backgrounds. And these individuals and their own mentees continue to push back the frontiers of science, Joanne style. Congratulations, Joanne. I wish I could present you in person.
Hi, everybody. My name's Joanne Stubbe, and what I want to do today is tell you a little bit about myself and then tell you how I've connected over the years with the chemistry department at Northwestern. I've always loved detective stories, the search for pieces to the puzzle, putting them together to solve mysteries. I've always enjoyed reading about the discovery of others. Detection and discovery, linked to my curiosity and persistence, had suggested that science was my calling. I was trained as a chemist in the late 1960s, and since that time, my science has always been interdisciplinary. I've held appointments in chemistry, biochemistry, and even pharmacology departments. My research has focused on understanding basic mechanisms of enzymatic catalytic transformations in biology, and their translationally important targets. My most recognized work is on ribonucleotide reductases, RNRs, enzymes that make all the building blocks essential for DNA biosynthesis. They use unprecedented metal cofactors and unprecedented radical-base reactions. They are the target of small molecule nucleosides, gemcitabine and clofarabine, that are currently used clinically as cancer chemotherapeutics. I've had many interactions with people from Northwestern chemistry department over the years, and I'd like to talk about that a little. The person I've known the longest has been Rick Silverman. He and I were at Brandeis working in Abel's lab, who was the inventor of mechanism-based inhibitors and rational drug design. Rick, at that time, worked on fluorinated amino acids as inhibitors of pyridoxal phosphate requiring enzymes. And I chose that topic as his work was really the inspiration for my studies on fluorinated nucleotides that I designed and studied as inhibitors of ribonucleotide reductases. Rick and I share a love of mechanism, rational inhibitor design, and chasing down the real mechanism by which these inhibitors work therapeutically. In Rick's case, detection and discovery led to the blockbuster drug Lyrica. RNRs require unique metallo cofactors, as I mentioned before. I knew nothing about metals in the early 80s, and I did a sabbatical in Lippard's lab at MIT. There I met Tom O'Halloran, a Lippard trainee, and was introduced at that time to the bioinorganic community, including my favorite collaborator of all times, Brian Hoffman. I knew little about the spectroscopic methods required to detect radical intermediates, and Brian taught me so much. He was always willing and to share and discuss intently his ideas. Brian now collaborates with Joan Broderick, who was one of Tom O'Halloran's PhD students. She did a postdoc in my lab. Together, they have made recently creative seminal discoveries on radical SAM enzymes that I think is noteworthy for is of Nobel Prize quality. Amy Rosenzweig, as well as Tom, is a Lippard trainee. She brought protein crystallography into the Lippard lab and did a spectacular work on solving the structure of a diiron enzyme that converts methane into methanol of environmental importance. It has a protein structure actually similar to part of ribonucleotide reductase, and these similarities resulted in our collaborations over the years and our discovery of a new metallocofactor in ribonucleotide reductases. She continues to do beautiful studies on the more prevalent copper-dependent methane monooxygenase. Grace Kenny, a former undergraduate student who worked in my lab at MIT, just received her PhD from Amy in the last few years. What a small world. Brian, Tom, Amy, and Rick, creative and passionate scientists, have always openly shared their ideas and their discoveries. They have made science really fun for me, and I'm grateful to them, and I'm grateful to Northwestern for this very special honorary degree.